CRISPR-Cas9, commonly shortened to just CRISPR, is one of the most powerful tools to come out of the last decade of biology research. But what are people using it for, and how does it work? CRISPR is a gene editing tool that researchers can use to edit DNA relatively quickly and inexpensively, and there has been exponential growth in its use since it was first used to edit DNA in 2012. The term CRISPR is an acronym for Clustered, Regularly Interspaced, Short Palindromic Repeats, which comes from its initial discovery as a defense mechanism in bacteria. In bacteria, CRISPR sequences are DNA sequences that contain many repeats of the same short sequence interspaced with so-called spacer sequences. Scientists discovered that these spacer sequences are actually copies of pieces of DNA from viruses that the bacterium has previously been infected by. Because the bacterium has captured and integrated this viral DNA as a spacer sequence, it can quote-unquote remember this virus. If it is infected by the virus again, it can recognize it and defend itself more quickly. It's kind of like the bacterium has a wanted sign for a known criminal. In this defense mechanism, once the bacterium recognizes the virus, it uses proteins called Cas proteins to cut up the DNA of the virus. Once its DNA is cut up, the virus can't infect the bacterium, so it's harmless. In recent years, scientists have discovered a way to use this same find and cut mechanism found in nature to edit DNA in the lab. This allows for easy gene editing. Essentially how it works is that scientists decide what part of the DNA they want to cut and design an RNA molecule with a matching sequence. They then attach this so-called guide RNA molecule to a protein called Cas9, whose job it is to cut DNA where the guide RNA finds its matching sequence. Once the DNA is cut, either a piece of DNA can be deleted or a new piece of DNA can be inserted. In practice, this is done either in cells in a dish or in embryos with only a few cells. In order for the organism to grow into an organism in which all of the cells have their DNA edited, it is necessary for all of the cells in the early embryo to have this desired mutation. If only some of the embryonic cells carry the mutation, the organism will have mosaicism, which means that the organism will have cells of two genetically different types, kind of like a piece of mosaic art. CRISPR has the potential to help us solve a lot of problems in biology and medicine, ranging from modifying crops to be more nutritious to eliminating genetic diseases such as cystic fibrosis and hemophilia. CRISPR is also an important tool in biology research and has enabled us to answer thousands of important questions. But when is it okay to use these technologies? Groups of scientists periodically get together to discuss this bioethical problem, and for now have decided that we're not ready to make CRISPR humans. The unknown risks are just too great, and it brings into question what parts of the human genome are and are not okay to change. Additionally, any changes made to a person's DNA in this way could be passed down to their children, and these mutations would enter the human gene pool. Nonetheless, there was a report this year that the first human babies to have their DNA edited using CRISPR have been born, much to the shock of the scientific community. It's important that we understand how CRISPR works and the potential that it has in advancing our society, but we must proceed cautiously and not inadvertently put people at risk. Thanks for listening. What topic do you want explained next?